Okay, so let's begin. This is renal anatomy. We can see we have the kidney right here. I'm just gonna draw to make it pretty evident what I'm talking about. So let's just start. We have the kidney right here. I'm gonna label that. The kidney is divided into two anatomical regions in a way. So you have the cortex, which is the outer layer. So it's gonna be hard to tell, but the cortex is usually the outer layer, and the medulla is the inner layer. And that kind of applies to most anatomical structures where the medulla is like the middle or inner layer, whereas the cortex is outside. You can see that in the heart, in the brain as well, sorry. Now moving on, we can see some other major anatomical structures here before we go into the kidney's anatomy. You have the bladder right here. None of this is really anatomically accurate, but this is just a conceptual kind of diagram. We have the ureters here. I didn't replicate the other kidneys, so we only have the right kidney for now, but these are both the ureters. And then finally here, arising from the bladder, we have the urethra. Okay, and this is just an external orifice, and that's urine. The kidney also receives blood supply from the abdominal aorta, which is, I don't think you really need to know, but just good to have. This is the renal artery. And this is the renal vein. And they're both pretty important because they divide into some smaller vessels, which I'll go into later. So these purple things we see right here, these are called the renal pyramids. And they play a pretty big role in feeding in the urine into the other structures I'll describe later. So this is called a minor calyx. And you can see the calyx, the minor calyx, is where the renal pyramid empties into. That's of one renal pyramid, and now you can see there's kind of like a kind of a crossroads point here. I'll make a mark right here. This is the major calyx, and it's pretty simple to understand. The minor calyx is just one, the major calyx is multiple. It feeds into from the minor calyces, and where all these major calyces kind of meet is what's called as the, I have to draw this all the way out here. This is the renal pelvis. And the renal pelvis is basically the beginning of the structure that we call the ureter, like I described earlier. The ureter just takes urine from the kidneys and brings it into the bladder. So just going from the top, the kidney filters the blood and creates urine. And that urine enters the minor calyx, then the major calyx, then the renal pelvis. The renal pelvis then empties or narrows into the ureter. And this is all happening on both sides because we have two kidneys in most cases. The ureters feed into the bladder, and the bladder empties into the urethra and then out of the external orifice. And that's the urine that gets excreted. Okay, so that was the anatomy. I'm going to go now move on to some logic because I think it's really important to understand why we produce urine in the first place. So why do we need a kidney in the first place? Why don't we just pee out blood, or why do we need to excrete anything at all, right? Because we could just keep cycling the same blood over and over again, and the heart will oxygenate it. So why do we need it in the first place? It's a good question. So every day we have muscle, and that muscle gets broken down. And we eat dietary protein that gets broken down. So we have all this nitrogenous waste that needs to be gotten rid of, right? So we can't just store that. It can't be recycled. It needs to be excreted from the body. Similarly, we have drugs that people take in the case that people are taking some kind of pharmacological substance or even recreational drugs. These kinds of things need to be metabolized and gotten rid of as well. And that metabolized waste can be recycled. It's pretty much useless to the body. So how do we get rid of that? We get rid of that through urine and excrement. But in this case, we're going to talk about the urine. So that waste needs to be removed. And moving on, why can't we just pee out blood? Because, blood has all this. Why do we need a filter organ at all? And I think that's also a pretty good question. But the important point to note here is that blood has some pretty useful things, and just because blood has some bad things, it doesn't mean we need to get rid of it in itself. We need to the goal is to really filter out the bad and keep the good. So let's just talk about some of the good things. We have red blood cells, which are pretty important for delivering oxygen to our body. We have sodium and potassium, and as you may know, they're pretty important for neuronal and muscle function. And they're also pretty important to keeping the osmolarity of the blood. And osmolarity just pretty much talks about how many osmoles, how many solutes do we have in our blood, and that pretty much defines how much we're going to have in terms of our fluid volume in our extracellular fluid and our intracellular fluid. So it's important to have a good balance of all these things to make sure we have an appropriate fluid volume. So we don't want to just get rid of sodium and potassium. Okay, so the next thing we don't want to get rid of glucose either, because glucose is really important to maintaining energy. And I didn't bring this up, but also amino acids and things like that are also important. We have things like albumin and proteins, and I'll talk about that a little later, but they're really important to maintaining the fluid volume and creating something called the oncotic pressure, and other important proteins in general. So we have lipoproteins, which transfer our lipids, and other various important proteins, and cells as well. So we don't want to get rid of these things, and water as well. We want to make sure we have an appropriate fluid volume. So the main goal, to simplify it really easy, is going to be pee out the bad things and reabsorb the good things, because we want to make sure this is as efficient as possible. And over time, the human body and biological systems have evolved such that the excretory system and the renal system, the kidney, work as efficiently as possible to get rid of things that are pretty much useless to the human body and reabsorb things that are really important to the human body. So let's take a look at some of the physiology stuff and get down to that pretty more in detail. Right, so this is a nephron. I didn't label it because I want it to be labeled so it'd be easier to go through. So I've added some of the functions that each part of the nephron does, so I think it's important to go over. So this right here, this is Bowman's capsule, this kind of Pac-Man looking thing, right? And it's holding the structure called the glomerulus. I'm writing that down here. And Bowman's capsule is pretty much the start of the nephron. And the glomerular capillary is not really, it's a capillary, right? It holds blood. So I wouldn't really consider it to be pure kidney tissue, whereas the Bowman's capsule is where I think the real kidney tissue begins. Not really important, but I think it's just a good way to understand what parts of the nephron correspond to what functions. So the glomerular capillary holds the blood like any other capillary would, and it's the smallest kind of unit of the circulatory system, going from artery to arteriole. So the glomerular capillary is just a small set of vessels that filters out blood, and we'll go into what that really means in a little bit, but I just wanted to include that to say what that kind of really does. So now moving on, after the blood from the glomerular capillary enters Bowman's capsule, it enters something called the proximal convoluted tubule. I'm going to write that here. You can abbreviate that to PCT. And from there, and in PCT, there's some really important functions that happen, including reabsorption. So without it, we would be losing a lot of the good stuff like I was talking about earlier. We want to reabsorb that good stuff to not lose it and be efficient with how we process our fluid. So entering from that, it goes into what's called as the loop of Henle. That's like a U-shaped thing, and it's pretty interesting in how it works, and I'll go into that later as well. So going here, we have our descending loop of Henle, and maybe I'll write it sideways. And over here, we have our ascending loop of Henle. So finally, from the ascending loop of Henle, we reach what's called the distal convoluted tubule, also known as the DCT. And from the distal convoluted tubule, we enter what's called the collecting duct. And that's kind of the final stage of urine formation in the nephron. Collecting. And from the collecting duct, just to go back to the anatomy, we would enter into the collecting system of the renal pyramid, going into the calyces and so on, so forth. So now we understand kind of the microanatomy of kidney tissue and renal tissue, and we also understand some of the larger structures. So now we can head into the actual specific functions and the physiology of each section of the nephron. So always to remember, the goal is to pee out some of the bad stuff, or all of the bad stuff ideally, and resorb pretty much all the good stuff. It's not perfect, but we've evolved significantly to allow ourselves to get as close to that as possible. So we'll start with the glomerulus and Bowman space. So let's just start with that first question. How does it help with the goal? So, the first goal to, peeing out bad stuff and reabsorbing good stuff is to make sure we just dump as much as we can out. And that's called filtration. So this is just a small image of the glomerular capillary and in Bowman space. And we can see that they're both kind of fluid-filled structures. Bowman space has a fluid inside of it, has some fluid inside of it, while the capillaries contain the blood. So what really happens is we have plasma leaving the glomerular capillaries and entering Bowman space. You might ask how that happens. And, it's pretty much all physics, fluid dynamics to some degree. So we can see how it's dumped out, and I think this is a really, really good fundamental equation in physiology. It's called Starling's equation. Probably not that important for the MCAT, but good to know, and the equation I think will definitely help. I can go into what these terms really mean. So GFR means the glomerular filtration rate. So if you think of this glomerular capillary as something that filters something out, a really important thing, and this is important clinically as well as it is philosophically and fundamentally to understanding the physiology. So the GFR is basically just the rate of fluid leaving the glomerular capillary and entering Bowman space. And that's across all the nephrons within the kidney. So if we look at the GFR, it's pretty much determined by three terms: the hydrostatic pressure and the oncotic pressure. So the hydrostatic pressure is just basically, if you imagine the glomerulus like a hose, the pressure that the fluid within that tube exerts on the walls of the tube is what we call the hydrostatic pressure. So if you think of a capillary, the pressure that the blood exerts on the walls, so it pushing out on the walls, is what we call the hydrostatic pressure. Same thing can be said about Bowman space. Bowman space isn't really a tube, but in itself, it's like a container of sorts, and any container will have what's called as the hydrostatic pressure. So, we have the hydrostatic pressure in both ends. So the hydrostatic pressure pushes in on the capillary. So fluid from Bowman space wants to enter the capillary, but fluid from the capillary also wants to enter Bowman space. So that's why we subtract the hydrostatic pressure of the glomerular capillary. We subtract the Bowman space hydrostatic pressure, from the hydrostatic pressure of the glomerular capillary. So PGC minus PB. Finally, we have another thing called oncotic pressure. And oncotic pressure is something that we can't really understand as intuitively. It's something that needs to be understood with some kind of experimental stuff helps you really think about it. And I've drawn something right here that could probably help. So if you look at this kind of container here, we have, you probably have done this in high school biology and chemistry. You keep a potato in a very, very hypotonic solution, like just straight plain distilled water perhaps. And after maybe a few hours or a few days, I'm not really sure about how long it takes, but we have water going into the potato. And the reason that happens is because that hypotonic fluid needs to enter the impermeable membrane that the potato represents, because all the stuff in the potato can't leave the potato because the skin, I guess, is preventing it. So what actually allows for the osmolarity to some degree, or the oncotic pressure to equilibrate in this case, is going to be our water entering that potato. So over a few hours or a few days, whatever that is, the potato becomes larger. So fluid goes in. So Bowman space doesn't have any protein. So note that, in this little schematic of the glomerular capillary and Bowman space, I haven't added any of these albumin dots. Albumin is like the largest or the most common protein in our body. So in our blood vessels, at least, not in our body. That's probably collagen. But there's no albumin proteins in Bowman's space. So there's only oncotic pressure going one way, which is pretty important to note. So if you look at the kind of distribution of the forces, we have pressure going out of the glomerular capillary, which is the hydrostatic pressure, and we have two sets of forces pulling water or fluid in to the glomerular capillary, which is the hydrostatic pressure of Bowman's space along with the oncotic pressure. The oncotic pressure, again, is only pulling fluid into the capillary because protein exists in the glomerular capillary and not in Bowman's space. So that's kind of why we see two sets of forces going into the capillary and one going out. But if you're talking about magnitude, how large are these forces, you're going to see that the hydrostatic force of the capillary is pretty much stronger than that of Bowman's space and the oncotic force or the oncotic pressure. And that's just because, the capillaries have a large supply of blood and they contain a lot of force. That blood exerts a lot of force on the glomerular capillary. So that's why we have filtration in the first place. Filtration won't happen unless we have more force going out than coming in, right? I think that's pretty intuitive to understand. So just in short, fluid pushes out and the albumin within the capillary pulls in. And we can see how that kind of affects the flow one more time by looking at Starling's equation. And if you want a positive flow rate, the only way that's possible is if PGC is way larger than, the oncotic pressure and of PB. Okay, so now moving on. What can't be filtered? And that's an important question because, these blood vessels are the capillaries are not fully just open to anything, right? Certain things can't leave, and that's protein and cells. So you're not gonna ever see in a healthy patient or in someone who's producing urine without any real problems with their kidneys. You're not gonna see red blood cells. You're not gonna see albumin. So if they're found in urine, that's an indication there's a problem. And it's not really important to know what problems, but I just think it's good to know because certain things just can't be filtered. And what can be filtered, so that's what we call plasma. And plasma is pretty much just blood minus the blood cells and protein. So it includes good stuff. I know we want to keep some of the good stuff and remove some of the bad stuff, but it's important to note that to start taking out bad stuff, you need to get rid of everything and then take back the good stuff so you can get rid of only the bad stuff. And I can show a good schematic of that here. So after we pretty much filter out the good stuff, we need to get that stuff back. So sodium, like I mentioned, important for our nerves and for our muscles. Bicarbonate is important because we want to maintain that pH. It's a biochemistry point, but I think it's important to note that every protein in our body is specific to a certain set of circumstances, and if the body isn't actively regulating our pH, then a lot of things will denature and then function will go. So bicarbonate is really important to reabsorb. Similarly, glucose required for various energetic functions, so that needs to be reabsorbed, and then amino acids as well reabsorbed. So we can see how all that stuff goes back. And it's kind of important to note that the albumin is at a bit of a higher concentration here because we lost a lot of fluid when we filtered. So now we have more of a force pulling fluid back into the glomerular capillaries, or not the glomerular capillaries, but the peritubular capillaries is what they're called after they leave the glomerulus. I'm going to write that down. It's not too important to note, but I wouldn't want you calling this the glomerular capillary. This is a peritubular capillary. And this right here is our PCT. So I'm going to go back up. This is going to be pretty fast. Our PCT is the first part we get out of Bowman's capsule. So the goal right there is to reabsorb the blood. Okay, so now that we took back some of the good stuff, I think it's important to know how we kind of change concentrations of the fluid within the nephron. And that's pretty much what the loop of Henle's about. So it's that big U, and each part of it. So this is another good thing to add. Talking about the anatomy of the kidney again. The outer part is the cortex. The inner part is the medulla. So at the top of the kidney, the cortex is a little more dilute, whereas you go into the medulla, you have a lot more concentrated fluid. And just like the Bowman space had fluid, we have something called the interstitial fluid that's right outside the tubules. Interstitial, not tubules, apologies. Interstitial fluid. And that fluid has an osmolarity. So the osmolarity of that fluid kind of determines, are we losing water or are we gaining concentration within, the tubular fluid, or are we, pumping it out? And I'll explain why that happens. So when we get the fluid out of the out of the glomerular capillary and into Bowman's capsule, it's pretty much about the same concentration as fluid in the blood. So there's no real change, there's no osmolarity difference, there's nothing like that. The way we change that fluid is by going in from the cortex into the medulla. So, like I said, the cortex is a lot more dilute than the medulla. So as you go deeper and deeper into the medulla, you reabsorb water, and that's very important to note. In the descending loop of Henle, it's permeable to losing water and impermeable to losing solute, like salt. However, in the ascending loop of Henle, it's permeable to losing salt, like I have NaCl here, leaving the ascending loop of Henle, and impermeable to losing water. There's a really important reason why that happens. So, water is pretty important to conserve. Like I was mentioning, blood volume and maintaining that is very important for the body. And there are a lot of checks and balances to maintain that. And this is something that's integral to renal physiology. So, we reabsorb that water, and it does two things. It not only saves some of the water and brings it back to the blood, but it also really, really concentrates the fluid within the nephron. And that's important for another reason. That if we concentrate the fluid within the nephron, it allows us to pump out that fluid on the ascending side of things. And what does that really do? It regenerates the gradient that I was describing earlier. So this whole gradient of cortex to medulla that I'm drawing this arrow right here, that's generated by creating a very concentrated fluid through this loop. And as we concentrate the fluid, there's more of a reason for salt to now be pumped out. This is very active transport. I'm going to write that down because it's important to note. Active transport. Right? So as we pump this out, we're pretty much adding into that gradient. As you go down, right, I've written some numbers. At the top, these are just estimates. I'm not 100% sure this is true, but at the top, it's around 300 milliosms. And as we go down, we go to about 1200. The tubular fluid, the fluid within the loop of Henle, is lagging, right? So it's not going to follow it immediately. It requires water to leave for it to follow. So when it's at 600 outside, it only is about 400 inside. And the only way it gets more and more concentrated is if water leaves, right? I think that's pretty easy to understand. If our concentration, and maybe I can write some small calculations here and erase it for later, but if we have 2 molecules of solute within 10 milliliters of water, our concentration is about 0.2. But now if we decrease that 10 to 5, we're going to get that concentration doubled to 0.4, right? So as we lose water, we're increasing concentration of the fluid. Now that we have an increased concentration of the fluid, we want to now pump out sodium so that we can regenerate that gradient. And it does two things at the same time, and that's pretty much what we call as a countercurrent multiplication system, right? So as fluid goes opposite directions, because as we're going descending, we're going down, and ascending, we're going up, we're creating a gradient for water extraction. So if we don't pump out this NaCl here, we're not going to have that gradient to bring out water here, right? It helps on both ends. And if we don't bring out water here on the ascending side of things, if we don't bring out that water, we're not going to be pulling up the salt on the other end. We're not going to be pulling it out of the fluid and creating that gradient. So it's important on both ends. As long as we have the salt being pumped out on this end, on the ascending end, as long as we have that gradient, we're gonna be able to pull out the water on the descending end. I'll just erase all my water. Okay, so that's gonna be a little confusing, but I recommend watching this over again and looking at this diagram to understand how both sides contribute. And I can explain it one more time. I think that'll probably be better. So again, we have cortex to medulla. There's a gradient, as it's called. It's more concentrated in the medulla than it is in the cortex. So as we put down the fluid, and as the fluid goes down into the medulla, there's a kind of force pulling water outside. And as we pull that water outside, the body conserves some of its water. That way we're not just peeing out liters and liters of water every day. So it's really important for that reason. And then as we go on this end, the fluid becomes insanely concentrated. As you can see, it's about 1,200 milliosmoles in the interstitial fluid, and I'm assuming it's going to be around that here as well, hopefully. And as we look at the ascending loop, now the fluid is more concentrated than the interstitium. So it might be 1,100 here, I don't know, it might be 700 here, it might be 500 here, 500 here. And that is what's allowing us to pull the NaCl out. And what that does is now we're adding solutes into the interstitium and making it more concentrated. That concentration now allows us on the descending end to pull out water once again. It's just a cycle. All it does is accomplish two things, and each side helps the other side do its job. Pretty much, a very, very synergistic system in a sense. And I think you just should really go through that over and over again so you can see how ascending helps descending and then descending helps ascending. Now that we've finished the loop of Henle, I think the final stretch is the collecting duct. And the collecting duct is really the place where we have a lot of hormonal regulation of things. So now we've pretty much taken out and brought back all the good stuff. How do we make sure we can get the urine out while not peeing out liters and liters of water? Because the loop of Henle definitely takes back some of the water, but also we can get some more water later. And that's pretty much at the collecting duct. So at the collecting duct, there's a bunch of things going on. There's sodium going from the collecting duct, and I'm just going to label the collecting duct because I think it might be important to remember that. Sodium leaves the collecting duct and enters the blood. And water also leaves the collecting duct and enters the blood. And you might remember from some kind of cell biology classes and understanding cell biology that, water can't really easily pass through the cell membrane because it's water. The cell membrane is filled up with lipids. So, you can't enter a hydrophobic area with water. So we need a channel, and that's what these aquaporins do. The thing about the aquaporins, though, is that they're highly subject to regulation. So, if we don't have this molecule or this hormone called ADH, or another name for ADH you might see on the exam is vasopressin, right? So if we don't have ADH or vasopressin, there's not going to be as much water re-entering the blood, right? So it's important to understand how that molecule and that hormone regulates the blood volume and the concentration of our blood and our urine. I'll go down and I'll explain a little more. So ADH is pretty much used when we're in that situation. We're stranded in the desert. We don't have water. We have a lot of solutes and waste being produced, and we're not really able to replenish that with water. So the concentration of our blood goes up. How would we want to bring that down, right? So just another example, I've drawn it out, a little container here with 4 pieces of whatever solute we have, or whatever 4 molecules, and it's 20 milliliters. So that suggests that our concentration is 0.2. And let's say in this system, 0.2 is really high. We want to bring it to 0.1. Without removing 2 of the solutes and just dividing it out down to 0.1 by taking out 2, we can now increase our volume and do it that way, right? So, because, the body can't just remove salt in and of itself. Salt in and of itself will just crystallize, and now you're going to get a kidney stone. So that's not a way to do this. We need to increase the water content of our blood to decrease the concentration. So that's what we've done here. We've added 20 milliliters of water to that initial container and brought down our concentration by half. So similarly, ADH does the same thing. If we want to lower the osmolarity, we need to increase the amount of water in our blood. So how do we do that, right? So going back up, aquaporins are upregulated by ADH or vasopressin, whatever you call it, to increase the amount of water in the blood. And now when we have high osmolarity of our blood, we can now decrease that by reabsorbing more water. Another hormone that's really important, possibly even more important, is called the renin angiotensin aldosterone system, RAS for short. So RAS is activated when sodium and water are low. It's a low volume state. So, I don't know why I spelled aldosterone wrong here, but the kidney has these detectors, these pressure detectors and salt detectors in it. And, they have names, but I don't think it's that important for you. So they just detect when there's low sodium and low blood pressure, and they release a hormone called renin. Renin is the first hormone. This RAS system is like a bunch of hormones released sequentially that do kind of the same thing. They have the same goal. They want to increase the blood volume and increase the sodium content. Because sodium pulls in water, because of its osmolar effects, it pulls in water. So it does both of those things, right? So we detect low sodium, low blood volume. We get renin released from the kidney. Renin converts this pro-hormone angiotensinogen. And important to know, anytime you see this word, something ending in an ogen, we know that it's not in its final form and needs to be activated. So renin converts angiotensinogen to angiotensin 1. Angiotensin 1 in and of itself, I'm not familiar that it does anything really crazy, but it gets converted by an enzyme called ACE, angiotensin converting enzyme in the lungs, to something called angiotensin 2. And angiotensin 2 has a couple of effects, so I'll go into those a little bit later. But angiotensin 2 now stimulates the production of a hormone, and it's a mineralocorticoid, to be very exact. Corticoid. It's found in this little purple thing I drew here. That's going to be our adrenal gland. And that produces things like cortisol. It produces, our sex hormones, testosterone, estrogen, and it produces aldosterone. I'm going to put a dotted line because I don't want this to take too much focus out, but aldosterone. And aldosterone does a lot of work in taking in salt and H2O. So to go back up to the collecting duct diagram here, the aldosterone activates, and I put RAS, but it's really aldosterone that does this. Aldosterone activates these sodium channels called ENaC, but that's probably not too important, but it activates sodium to bring back into the blood, and that sodium pulls in water with it. So what ends up happening, I'll go through the loop one more time. We detect low sodium or low blood pressure. Renin gets activated. Renin converts angiotensinogen to angiotensin 1. Angiotensin 1 gets converted by ACE to angiotensin 2. Angiotensin 2 stimulates aldosterone production by the adrenal gland, and aldosterone, to go up one more time, activates sodium reabsorption, and with that sodium we get some water, and now we increase our sodium and water, and now we increase our blood pressure. So in a low volume state, RAS does the job to bring us back to some level of normal. I was talking about angiotensin 2 as well earlier, and I wanted to explain what it does as well outside of its effects of activating aldosterone production. Angiotensin 2 also affects filtration, and this is a different kind of model of the glomerular capillary and Bowman space. So we have the afferent arterial and efferent arterial. I didn't describe that earlier, but I think it's really important to note. Similar thing to the nervous system, the afferent arterial is just what's going in, and the efferent arterial is what's coming out. So we have, again, the Bowman space right here. Women's capsule, we have the glomerular capillaries. So again, I was talking about GFR earlier, and there are a couple of things that determine GFR, not just our hydrostatic pressure and our oncotic pressure, but there are things that affect the hydrostatic pressure of the fluid within that glomerular capillary. So this is kind of like talking about the radius of a hose, and I drawn a bit of a hose here to explain how this happens. Angiotensin 2 constricts the efferent arteriole, and that increases our GFR. So to think of it like a hose, to think of our capillary like a hose, let's just imagine a hose with the hole in it. There's not only the part where water leaves the hose, but then proximal, right, more before that final hole, we have a little, breakage in the hose. It's a very old hose. Let's imagine it like that, right? So when we have a normal hose with no kind of constrictions in it, there's not really that much water leaving through that little hole behind. Let's say we have a clamp now, and we clamp it down. We're going to have a lot more water leaving through that hole, and not as much leaving through the actual exit point of the hose. I've drawn that here. You can see these large arrows here symbolizing that there's a lot of water leaving prior to the clamp. And that's basically what we have in the glomerulus, right? We have the efferent arteriole being clamped down on, and that allows for more to be filtered out. I'm going to draw these arrows here showing how we have more filtration. And that's pretty much the same thing as the hose. I've drawn what each part represents as well in the hose. So we can see right here, the proximal represents the afferent arteriole, and the distal part represents the efferent arteriole. The clamp is basically what angiotensin 2 does, and the high flow is what's equal to a high GFR. So finally, the takeaway I want you to get from this is that angiotensin 2 not only allows us to increase our blood volume via activating aldosterone production in the adrenal gland, but it also allows us to increase our filtration rate. Okay, so these are some final points. Couldn't really fit them in other places, but the kidneys play a really important role in regulating the pH of the body. Like I was talking about earlier, bicarbonate is very important as a buffer, and pH is important to determining the structure of our proteins because we don't want them denaturing. We want to maintain the function, so pH is really important to regulate. So when the body is acidotic, when our pH of our blood is very, very low, the kidney just pees out a lot of protons. That allows us to get rid of some of that acidic material, and it allows us to now increase the pH of our blood because we're getting rid of the protons. Similarly, when we're alkalotic, when we have a really high pH, the kidneys excrete bicarbonate, and now we're bringing ourselves back to that set point and that normal. Okay, so another point, I was talking about glucose earlier. We try to reabsorb as much glucose as possible, but that's only to a real limit. So when the kidneys can't reabsorb as much is when we get into some problems. And when we can't reabsorb, when we're kind of saturating the amount of reabsorption, is when we have something like diabetes. When the blood glucose is really, really high, we're not going to be able to reabsorb as much as we filter. So now we get glucose in the urine. And maybe I can just draw this out because I think it's important to remember this. We filter stuff. The amount we excrete, right, is equal to the amount we filter minus the amount we reabsorb plus what we secrete. That's important to note. So if we think of the tubule contains some material in it, and just draw the fluid. Whatever ends up going out of our orifice, or whatever leaves our body, right? Whatever leaves our body is what's excreted. Whatever re-enters is what's reabsorbed. Whatever's pushed in from the blood is what's secreted. A lot of things like drug metabolites, sometimes protons when we're really acidotic. These are kinds of things that are secreted, that we just keep pushing them into the urine. Finally, from our opening, or I'll do that in the yellow to correspond. That right here is what's filtered. This is what's secreted. So this equation is pretty easy to understand what's going on, right? Whatever we end up excreting is equal to what we filter in the first place. We take some stuff back, and then we actually secrete a bunch of other stuff as well. We want to get rid of the stuff we secrete. We want to keep the stuff we reabsorb. All goes down to that final goal of, pee out the bad stuff and retain the good stuff. So the final point I want to make is that mammals, we excrete our nitrogenous waste as urea. And that's something that's filtered. It's not really reabsorbed much. So the body tries to make sure that we filter out the urea and other nitrogenous waste because, it's not really that useful to the body. We don't really do much with that. So with that, that's pretty much what I have for, renal, and I hope this is of value to you.